This mutualism between heterotrophic mycorrhizal fungi and autotrophic plants is only one example of a symbiosis between fungi and plants. Another example is called Mycoheterotrophy. In this case, the plant isn't an autotroph. It doesn't have chlorophyll and it doesn't have chloroplasts. Instead, the plant is a heterotroph, and the mycorrhizal fungi that it is in association with provides sugar to the plant. Now, the same mycorrhizal fungi that these mycoheterotrophs grow on is also connected to root networks of autotrophs. And so you have a flow of sugar from an autotrophic root system through some mycorrhizal fungi to the mycoheterotrophic plant. Now, these mycoheterotrophs are found in many different lineages, but in particular, I'm going to talk about two examples from Monotropoideae, really just because they kind of look cool. On the left here, we have Monotropa uniflora, and on the right, we have Sarcodes sanguineae. Both of these, obviously, are completely devoid of any green coloration, and these Sarcodes sanguineae are actually extremely bright red. And so they don't have pigmentation that allows photosynthesis, but this pigmentation is still beneficial if these plants are going to be out in the sun. And so these Sarcodes sanguineae are often some of the first plants that peek up through the snow in the spring. And this really high level of pigmentation helps protect it from UV damage and oxidative stress in general. On the other hand, the Monotropa are really kind of dim and pale because they often live in the shade. And so they're at an advantage because they don't need sunlight, and actually producing a lot of pigment would be a disadvantage in that case, just because it takes energy to produce it, and then there's not going to be a lot of UV where that pigmentation would be an advantage. Now, as I mentioned, both of those species of plant get sugar exclusively from mycorrhizal fungi. And these mycorrhizal fungi can be either ectomycorrhizal fungi or endomycorrhizal fungi. Endomycorrhizae, as you can tell from the name, penetrate cells and actually end up on the interior of the plasma membrane, whereas the ectomycorrhizae penetrate the epidermis of the plant, but then the hyphae grow between the cells as opposed to into the cells. Now, when these mycorrhizae grow in association with the Monotropoideae, they're called Monotropoid mycorrhizae. In the top left here, there's a diagram of one of these associations between an ectomycorrhizae and a Monotropoideae plant. In this case, it's this Monotropa uniflora. And so this M layer is called the mantle, and that's made of condensed mycelium. The H is this so-called Hartig net. That's hyphae that are actually in contact with these cortical cells of the plant. From here, the hyphae are going to grow in between these cells, and that's going to give them access to the phloem and the different tissues of the plant to facilitate the movement of the sugar into the mycoheterotroph. Now, in the case of these Sarcodes sanguineae, the structures are going to be similar. You're going to have these nodes on the root tips, but the particular fungi that grow in association with the Sarcodes are going to be different species and possibly even different families of fungi compared to the ones that grow in association with these Monotropa. It's generally the case with these plant-fungus associations that it can be oftentimes even species specific, and so the plant will give off some sort of exudate that attracts the fungus. The fungus then comes into contact with the root tip, and from there it begins growing through the epidermis and either into or between the plant cells. And just to sum it up, I think these mycoheterotrophs and similar symbioses are a really good example of the hidden and sometimes hard to see diversity and complexity in ecosystem interactions.